So we spoke about the genomics of cancer, which is essentially how people thought of cancer since people know of cancer. So it was always thought that you have mutations in some gene, which would be either oncogenes or tumor repressor genes, and like some there could be like a nucleotide mutation, or the whole genome, like the whole gene can get transferred from one chromosome to another chromosome. So there are essential chromosomal abnormalities or mutations which will lead to cancer. So this is a sort of new idea which is becoming more and more prominent, and it puts epigenetics and epigenomics at the forefront of cancer research. So I took this image from Economist, and it was a really nice article, and actually I took the title also from that Economist article. And what they talk about is like so essentially, like Ram spoke of okay, yeah. So Ram talked about the genomics, basically changing the gene for the cancer. So I'm going to take the epigenetic approach and essentially answer two questions, which is epigenetics at all important in cancer research, and what we as like Ha and Myong group members can do to target, like use epigenetics as a tool to target cancer. So just to begin with, I'll first define what is epigenetics or epigenomics. So epigenetics is anything which involves something over your DNA, and it doesn't involve any changes to the DNA mutations. So essentially, people talk about four different aspects of epigenetics, which together form epigenomics. And so first is DNA methylation, and this is the most important and the most well-studied cause of cancer when we talk about cancer epigenomics. And what happens is, so it's known that mammalian genome has these cytosine and CpG islands which are non-methylated. And these CpG islands, when get methylated, there is a silencing of certain genes. And then these genes, people have, I'm going to talk a little bit more about this, but people use these two sorts of techniques, bisulfite sequencing and DNA immunoprecipitation, to determine the regions of methylation on the DNA. The second epigenetic modifications, epigenetic changes which take place in our genome are histone modifications. Essentially, the histones, as Thui pointed out, are made up of like 4 different types of histones which form the nucleosome, and your DNA is wrapped around it. So you can imagine, like, if the DNA is wrapped around it, it would be blocked for like any sorts of replication or transcription process. And there are marks on these histone tails which are very well studied for activating and repressing the genes. And people study them using chromatin immunoprecipitation. Another not so well studied aspect of cancer epigenomics is non-coding RNAs. I would briefly touch upon this. And then the fourth aspect is the chromatin structure itself, like how the chromosomes are arranged in the nucleus and the 3D arrangement. So this is essentially studied by 3C, 4C, and high C methods. So what you can imagine in cancer to take place, like how the epigenetic can change like cancer cells. So essentially, people talk about 2 methods. First is you do some heterochromatic changes. So what would happen is, like, the histones which are present near your genes would get activated or repressed. So they could activate an oncogene, or they could repress a tumor repressor gene, and would cause proliferation of cells and lead to abnormal cell division and cause cancer. And the second aspect is DNA methylation, which is essentially putting methylation marks on the cytosine residues, and which would silence the genes. So these are essentially known as tumor repressor genes, and would cause, again, abnormal cell division and lead to cancer. So people have idea about, like, so these things in concept can affect the expression and expression of certain genes inside the cell, and they can lead to abnormal growth. So there are two big consortiums which are trying to understand how epigenomics of cancer cell is different from normal cells. And this is NIH and this is Wellcome Trust. And so I'm just summarizing their results, but they have a lot of studies on different cancers and different stages of cancer. So what they do as part of the consortium is use these techniques I described to map the epigenetic differences, essentially these DNA methylation marks and histone modifications between normal and cancer cells. And then they are also trying to create a map of these epigenetic modifications in different types of cancers. So what their findings are, and it is quite consistent and people believe in it quite strongly. So cancer has epigenetic signatures. So there are, suppose skin cancer has certain genes which would be methylated and acetylated. So you can identify, if you look at the epigenome of the cell, you can identify it as cancer or not. Okay, and then the most important part for any sort of cancer cell that whatever changes happen to your cell should be heritable, because each cell will need to divide repetitively. So these modifications or these epigenome is heritable, which is also known independently from studies on normal cells. And then these modifications are actively lead to overexpression of oncogenes or silence the tumor repressor genes. So the genes themselves do not have any mutations, but it's the regulation which gets perturbed. So what I'll spend another 2 minutes to talk about how in our lab we can study this. So a dream experiment which I would design is something like you have a group of cells and you want to identify which cells are cancerous cells. Okay, first of all, that's a major challenge. And so you need a sure shot way to identify, okay, this is a cancer cell. And I'm looking at epigenomic marks. So after you do that, you know it's cancer cell. So the red one's a cancer cell. And you want to devise some strategies to kill the cancer cells. So the good thing about epigenomics is that epigenomics marks are reversible. So you can do certain enzymatic reactions on the DNA and the histones and reverse these modifications and bring the cancer cell to the normal cell, which is not very easy with doing genomic manipulations or doing gene therapy, by using gene therapy. So this is one of the strong, I would say a fortunate property of epigenomic modifications in cancer. So I have heard about if we want to do these experiments right now, from when we go back, and what can we do? So talking about the first aspect, which is identification of epigenomic marks. So the idea is all these studies involve looking at lots of cells, and they cannot identify single cells, and as Jason mentioned, that you need to detect the cancer at early stage. So at early stage, each cancer starts with a single cell. So the idea is to look at a single cell, or at least few cells in number. And what we can do is essentially do some protocolization studies using, okay, I'm not going to go into detail of all these methods. I'm hoping you guys would know. Co-localization studies using some epigenetic marks. Essentially the idea is if I take a part of the genome or the chromatin, if it has, if it has active modifications, if it has repressive modifications, and how do they change from normal cells to cancer cells. We can also devise some in vitro FRET assays in the sense you can have a protein which is responsible for putting suppose methylation marks, like a methyl transferase. You can use some sort of FRET assays to actively monitor the methylation taking place inside the cell. So this is something which no one has done, but I think it will be pretty cool image giving live cell imaging of epigenetic modifications taking place. And so since these cancer cells need to metastasize and like go from their origin to a destination. So there are lots of cell matrix changes which are all mechanical. So we also have tools to study that. And another thing is these non-coding RNAs. So telomerase are responsible for, I was reading, like approximately 90% of cancers, which is somewhere here. And non-coding RNAs have been implicated as epigenomic regulators of these telomerase. So this is something which we can try using our methods by putting them on the surfaces. And on the side of like curing cancer, actually there are lots and lots of drugs which are available which would modify these epigenomic marks. And I'm listing 3 famous ones. And essentially these drugs target like DNA methylation. This is a methyltransferase. So the drug targets a methyltransferase and prevents the methyltransferase to put silencing marks on tumor repressor genes. And this is one of the both of these are FDA-approved drugs, and they are used for curing leukemia. And this is one of the drugs which is very hot right now, and it is supposed to block the overactivity of MYC gene, which is implicated in a lot of cancer. It's an oncogene essentially. And apart from that, there are lots of other drugs. I don't exactly know how many of them are actually in clinical trial or approved, but they target different proteins which are responsible for putting different sorts of epigenetic marks on the histone. So these drugs can be used to, can be given to the cells, and we can see how they change the epigenome of the cell. So that's what I'm going to talk about, and thanks to Vasudha for organizing this and the planning committee for taking care of everything. I hope you have a lot of questions.
All right, any questions?
So how are these drugs directed to specific cells?
Yeah. So most of the, what I read is so for epigenetic thing, just erasing the epigenetic marks, you can change it by changing the dosage. So the dosage of these drugs is very important. And essentially, they affect the cancer cell more significantly than the normal cells. So that's what the good thing about these drugs are. They don't exactly know how that works, but the idea is the normal cells are able to get the marks back, but the cancer cells die because the effect of the drugs on the cancer cell is much more than the normal cells.
Any other questions? All right. Oh, Judy?
So I have some people who say that another kind of drug that specifically targets cancer cell is the inhibitor, for example, PARP inhibitor. So
Yeah,
we have a more level of that PARP in cancer cell. So that drug only targets specifically the cancer cell. And so the idea of that drug is stopping the growing of cancer cell instead of fixing it.
Yes.
So do you know how it compares to the traditional drug that we use today?
So what these drugs do is essentially so if it's a DNA methylation drug, so what it will do is it would prevent DNA methylation from taking place in cancer cell. So what would happen is the genes which were getting silenced near to that DNA methylation would be active again. And they would be able to, so those would be tumor suppressor genes which were getting suppressed, right? So they can be active and they would prevent the growth of the cancer cell. And the cancer cell would eventually die.
Yeah, so